Welcome to the deep dive. We sift through the sources, pull out the key info, and, well, hopefully leave you feeling genuinely well informed, maybe even with a few surprises. Today, we're tackling an animal health issue, and at first glance, honestly, it might seem, well, almost minor. We're talking about a virus, low mortality rates usually, but don't let that fool you. This goes way beyond just a few sick animals. It's actually a national agricultural security concern. It triggers these immediate high priority responses. Think state, federal officials, sometimes even shutting down livestock markets.
It really is a fascinating paradox, isn't it? We're diving into vesicular stomatitis today, or VS. Now, this virus, it hits a pretty wide range of livestock, cattle, horses. But our focus, our mission today is really zeroing in on its effects in two, less studied groups. The Camelidae, so your camels, llamas, alpacas, and the Cervidae, which is deer, elk, moose.
Right, so the mission is to figure out why this disease, where most animals actually get better in what, a couple of weeks, why it can send these huge shock waves through the whole agricultural sector. We'll look at what causes it, how it jumps around, what it actually does to the animals, and crucially, we'll point out the big knowledge gaps. There are still things we just don't know, especially about those camelids and cervids. It all comes down to the virus itself. Vesicular stomatitis virus, VSV, these tiny insects that carry it, and this really weird, uncanny resemblance it has to another disease, a much, much scarier one.
Exactly. And to really get why VS is such a big deal, we have to start with the actual virus. The vesicular stomatitis virus, VSV, it's bullet-shaped single-stranded RNA, belongs to the Rhabdiviridae family. In the Americas, we're mostly dealing with two main types, or serotypes, New Jersey VSNJV and Indiana VSIV. And it's worth remembering that the New Jersey type, VSNJV, that's the one causing most of the trouble here in North America. The Indiana subtypes, especially 2 and 3, you see those more down in Central and South America.
Okay, so this bullet-shaped virus.
Yeah.
How does it get around? Is it mostly animal-to-animal contact or something else?
That's a really key point. So while direct contact can spread it later, the main way it initially gets transmitted is through vectors. We're talking tiny biting insects. Things like sandflies, black flies, and those little biting midges, chelacoids. They get infected when they bite an animal that already has it, especially near those blisters, the vesicles, which are just loaded with virus. Then when they go for their next blood meal, bang, they pass it on to a new animal.
Ah, okay. That makes total sense then with the timing we usually see, like late spring through fall, May to October, roughly.
Precisely. That's peak insect season, right?
Yeah.
And the outbreaks usually die down pretty quickly once you get that first hard frost.
Right. The cold kills off the vectors.
Exactly. And geographically in the U.S., you often see outbreaks starting down south, southwest Texas, New Mexico, Arizona, and then they tend to move northward, often following river valleys, which again lines up with where these insects live and travel. Now, the really interesting bit is how the virus survives the winter. We still don't know for sure. The main idea, the leading hypothesis, is that it actually overwinters inside the insect populations, possibly through something called transovarial transmission.
Transovarial, meaning?
Meaning the infected female insect might pass the virus directly into her eggs, so her offspring are born already infected. And that would explain why an area that had an outbreak one year is often at higher risk the next year. The virus might still be lurking in the local insect population.
That's a clever way for a virus to stick around. But it's not only insects, is it? Once it's in a group of animals, can they spread it directly?
Oh, absolutely. Once it's introduced, direct contact becomes really important for spreading it within a herd. The fluid inside those blisters is packed with virus, and saliva, especially from animals with mouth lesions, is highly infectious. So just nose-to-nose contact or even sharing things like feed troughs, water buckets, milking gear, what we call fomites, can spread it. The virus can actually hang around on those surfaces for, about 3 or 4 days.
Okay, we know the villain, we know how it travels. So what does this blister disease, as some call it, actually do to the poor animal that gets it? What are the signs we'd see?
Well, fundamentally, VSV attacks epithelial cells, the cell's lining surfaces. This causes cell death, fluid builds up, and you get these fragile blifters or vesicles, and they break really, really fast, often within just 48 hours, leaving behind these raw, very painful ulcers. The incubation period, the time from infection to signs, is usually pretty short, typically 2 to 8 days.
I heard the very first signs can be kind of subtle, though, maybe easy to miss.
Yeah, you might see a temporary fever, maybe up to 40 Celsius in cattle, but that can actually come and go before the vet even gets there. The most obvious first sign you'll usually notice is lots of drooling, just excessive salivation. That's because their mouths are incredibly painful. Then you see the classic lesions, the blisters and ulcers. They pop up in specific spots: tongue, lips, gums, the dental pad, muzzle, inside the nostrils, and crucially, you also see them on the coronary bands right at the top of the hoof. That causes lameness. And on teats and udders in Gary animals. Horses might also get them on their ears, sheath, or even their belly.
Ouch. That
Yeah.
sounds really painful. What are the bigger impacts on the animal beyond just the sores?
Oh, it's definitely not just a mild thing for the animal experiencing it. Think about it. Intense mouth pain means they don't want to eat or drink. So, anorexia, dehydration, they lose weight fast. For dairy cows or goats, those teat lesions make milking agony. Milk production plummets, and they're prone to getting secondary mastitis, which is a nasty udder infection. And the foot lesions, the coronary band ones. Severe lameness. In really bad cases, the inflammation can even cause the hoof wall to separate to slough off. Now, the good news generally is that the prognosis is usually good. It's self-limiting, the body fights it off. Most animals with some supportive care are fully recovered in about 10 to 14 days, and they develop really strong, long-lasting antibodies afterwards.
Okay, so that's the picture for the common livestock, but we wanted to really dig into these two other groups. Let's start with Camelidae, llamas, alpacas. They are officially listed as susceptible, right, by the big groups, USDA APHIS. Whoa.
Absolutely, yes. There's no question, officially. New World camelids, llamas, and alpacas are recognized as susceptible hosts by all the major international and U.S. animal health authorities. But, and this is the really strange part, the paradox, despite being on the list, actual clinical cases in llamas and alpacas are always described as occasional or rare, especially when you compare them to horses and cattle during the same outbreaks.
That is weird. So the rule books say they can get it, but in reality, we don't see it much. That's a big puzzle.
It's a central mystery, really. We do have records of them being investigated, like 4 camelids in 2009, 5 alpacas in 2012, 3 more camelids in 2015. They were all checked for suspected VS. So they're definitely included in surveillance efforts. But the problem is there's a real lack of detailed published studies, peer-reviewed case reports describing exactly what VSV looks like in a llama or an alpaca. Most of what we think we know is just borrowed, extrapolated from horses and cattle. Yeah. So why so rare? Is it that they're just biologically less susceptible somehow? Or maybe their housing or management means they just don't get exposed to the insects as much? Or maybe they get milder signs or signs that look like something else so it gets missed? We just don't have the answer yet.
So purely hypothetically then, if a llama did get VSV, what would we expect to see based on what we know from other animals?
Well, based on that extrapolation, you'd expect those vesicles, the blisters, turning into ulcers or crusty sores. Probably on the lips, muzzle, inside the mouth, especially the tongue. Maybe lesions on the coronary bands causing lameness, or on the teats if it's a breeding female. And the signs that go with that: drooling, not wanting to eat or drink, losing weight, maybe limping.
And are there any particular challenges when you're trying to diagnose something like this in a camelid? Their mouths are a bit different, aren't they?
They are, definitely. If a llama or alpaca is drooling or seems to have mouth pain, a vet has to think about other things too, like megaesophagus, problems with their stomach emptying, even just heat stress can cause drooling. The absolute key thing pointing towards VSV would be actually finding those typical blisters or ulcers. And yes, their anatomy makes it tricky. They have that split upper lip, and the back of their throat, the oropharynx, is quite narrow. So getting a really good look inside can be tough. Takes some skill.
Right. And the official response, the regulations, if you suspect VS in a llama or alpaca, I'm guessing it's just as serious as in cattle.
Maybe even more so. Oh, absolutely. You've hit a critical point there. The diagnostic steps are exactly the same. Any suspicion, you report it immediately. State vet, federal vet.
Yeah.
That triggers the official investigation. Sample collection swabs from lesions, blood samples taken weeks apart to look for rising antibodies. Treatment is just supportive care, but biosecurity, keeping things clean, controlling insects, that's vital. And because llamas and alpacas are cloven hoof, they have that split hoofs like cattle, sheep, deer. Any case with blisters automatically raises the alarm for foot and mouth disease. So yeah, rapid reporting and diagnosis are absolutely non-negotiable. The stakes are incredibly high for the whole country's FMD-free status.
Makes total sense. Okay, let's switch gears to our other group, Cervidae. Deer, elk, moose. This seems like an even bigger head-scratcher. Lots of evidence they get exposed, but almost no sign of actual sickness. How does that work?
That is the core disconnect, exactly. Blood tests, serological surveys, they consistently show antibodies to VSV in wild deer populations all over North America. Like one study in the southeast U.S. found over 2% of white-tailed deer had antibodies. And often, the places where they found positive deer were near areas where VS was known to be active in livestock. So white-tailed deer, yeah, they're widely seen as a wild host for VSV. They are definitely getting infected.
Okay, so the blood tests prove infection, but they just walk it off. It's baffling for a disease that makes other animals so obviously miserable with blisters.
It is baffling. Despite all that antibody evidence, finding a wild deer, elk, or moose that's actually showing clinical signs of VS
Yeah.
is almost unheard of. Documented cases are virtually non-existent. So why? Several ideas. One, maybe they usually just get subclinical infections. They fight it off, develop immunity, but never look sick. Two, think about how hard it is to even spot a sick wild animal. They're elusive. A deer feeling a bit off, maybe slightly lame, you'd likely never notice it out in the woods. And three, maybe if they do show mild signs like drooling, it gets mistaken for other common deer diseases. Things like adenovirus hemorrhagic disease or even chronic wasting disease can cause similar vague signs.
This leads to a really big question then. What role are these deer actually playing in the whole VSV picture? Are they just innocent bystanders getting infected, or are they reservoirs? Can they keep the virus going and spread it back?
That is the crucial unanswered question in VSV ecology. A true reservoir host needs to be able to maintain the virus and effectively transmit it back to others, like insects or livestock. For VSV, that likely means the deer would need to have enough virus circulating in their blood viremia or have active virus-shedding lesions to easily infect biting insects. But the experimental studies we have suggest deer tend to have only very low levels of virus in their blood and only for a short time before they develop strong immunity. This makes them seem like poor amplifiers of the virus. And if they mostly get subclinical infections without many lesions, well, their ability to pass the virus back to insects would be pretty limited.
So the current thinking leans more towards them being like a dead end for the virus.
That seems to be where the evidence points right now. More likely incidental hosts or spillover hosts. They live in the same areas as the insects and livestock, so they get exposed frequently. But they might not be very efficient at keeping the transmission cycle going strong.
Which must make trying to track VSV in wild deer incredibly difficult, right?
If they're not sick, how do you even monitor it?
It's a huge challenge. You're relying mostly on opportunistic samples, blood from hunters, maybe roadkill. It's not systematic. But here's an interesting idea that's being floated. Even if they aren't reservoirs, maybe deer could be useful ecological sentinels. Imagine setting up a planned, regular program to test blood samples from local deer herds. If you start seeing a rise in antibodies, that could act as an early warning. It could tell us that VSV activity is increasing in the environment, signaling a higher risk for nearby farms before their livestock start getting sick.
That's a really neat concept.
Yeah.
Okay, now let's hit the absolute crux of why VS matters so much nationally. It's not really about the disease itself being so bad usually.
Yeah.
It's because it looks exactly like something else, right? Something genuinely terrifying. You've nailed it. That is the #1 reason VS causes such alarm. Its importance isn't the morbidity or mortality, which are generally low. It's almost entirely down to its clinical mimicry looking identical to foot and mouth disease, FMD. FMD is caused by a different virus, an aphthovirus. It's incredibly contagious, devastating economically, and it's a foreign animal disease. We stamped it out in the U.S. back in 1929, but the threat of it returning is constant.
Wow. So if a vet sees those blisters, those vesicles, especially on an animal with cloven hooves like a cow, llama, or even a deer, they must treat it as potential FMD until proven otherwise. That's immense pressure.
It's an absolute ironclad rule. Any suspect vesicular case in a cloven-hoofed animal is handled as potential FMD until lab tests give the all-clear. It triggers this immediate high-gear regulatory response. State vets, federal vets, diagnostic labs, everyone mobilizes to rule out that worst-case scenario, an FMD outbreak. The consequences would be catastrophic.
But there's one key difference, a sort of field clue that can help distinguish VSV from FMD involving horses.
Yes, that's a very important distinction. Horses get VSV quite readily. They are susceptible, but they're completely resistant to FMD virus. They just don't get it. So if you arrive at a farm and see both horses and cattle, or horses and llamas, showing similar blistery lesions, that strongly suggests VSV. But if you only see it in the cloven-hoofed animals, just the cattle, just the sheep, just the pigs, while the horses are fine, then FMD has to stay top of the list until the lab says otherwise. That horse clue is vital.
And the economic fallout from this look-alike issue, even if it is just VS, it's still huge, isn't it? It's not only about the animals being sick.
Oh, far from it. The direct costs are bad enough. The animal suffering, lost weight, lost milk, vet bills, potential lameness issues.
Yeah.
But the indirect costs are often much, much worse. Think immediate quarantines on farms, restrictions on moving any animals in the area, cancellation of shows, sales, fairs, major economic hubs, and then there's international trade. Countries we trade livestock or products with might slam the door shut, block imports from affected regions, even if it's only VS, because of that FMD scare. We've seen examples, like 2 dairies in California losing maybe $225,000 over just 2 months. Studies on beef ranches found average losses over $15,000 per affected farm. It adds up incredibly fast.
So, diagnosis, prevention, control, it sounds like this requires a really well-oiled system that's always on high alert.
That's exactly what it is. VSV is nationally reportable. Any suspicion, even just a maybe, must be reported straight away to the state or federal animal health officials. No delay. Then, a specially trained vet, a foreign animal disease diagnostician, FADD, they come out. They lead the investigation, collect the official samples, fluid from blisters, bits of tissue, swabs, and those crucial paired blood samples taken a few weeks apart for antibody testing.
What kind of tests are they running in the labs to get those answers, especially with the FMD clock ticking?
They throw a whole battery of tests at it. For detecting the virus directly, they can try to grow it in cell culture or use a ELISA test to find viral proteins. But nowadays, genetic tests like RT-PCR are really key. They're fast, super sensitive, and they can even run multiplex assays tests that look for VSV, FMDV, and other vesicular viruses all at the same time from one sample. For antibodies in the blood, they use tests like complement fixation, CF, virus neutralization, VN, or competitive ELISA. The goal is rapid, accurate confirmation and exclusion of FMD.
And the local vet, the one who first sees the animal, they're really on the front lines here, aren't they?
Absolutely critical. The field veterinarian isn't just treating an animal. In these cases, they're the first line of defense for the entire country's agricultural biosecurity. Their ability to spot suspicious signs, make that call immediately, and cooperate with the regulatory folks, it's absolutely paramount. Every investigation is a test of the system.
Since there isn't a widely used vaccine for VSV, what are the main strategies for stopping it once it starts?
Yeah, control is mostly reactive. It's about breaking those transmission chains, stopping the spread. The official response kicks in fast. The infected farm is quarantined, usually for at least 14 days after the last animal is healed. And often, wider movement restrictions get put in place for the region. Things like needing a health certificate, a certificate of veterinary inspection, just to move livestock across state lines.
And back on the farm itself, it really comes down to strict biosecurity and fighting those insects?
That's the core of it, yes. Since insects are the main way it gets introduced, vector control is huge. Physical barriers like screens on barns, getting rid of standing water and manure where insects breed, using big fans in stables because insects don't like strong air currents, and applying approved insect repellents or insecticides directly to the animals. Sanitation is just as vital. Isolate sick animals immediately. Have a strict order for chores. Always handle healthy animals first, then the sick ones. Clean and disinfect everything, feed buckets, water troughs, halters. Bleach works well against VSV. Avoid sharing feed or water sources between groups.
What about people? You mentioned vets need to be careful. Is there a risk for farmers or anyone handling these animals?
Yes, there is. VSV is zoonotic, meaning it can pass to humans. People usually get flu-like symptoms, fever, headache, muscle aches, just feeling generally unwell. It typically comes from direct contact with the animal's lesions or saliva. It usually clears up in maybe 3 to 5 days. Very rarely, someone might get blisters in their mouth. That's why personal protective equipment, PPE, is so important. Gloves are a must. Eye protection too, maybe a mask, for anyone handling animals suspected of having VS.
So for people with llamas, alpacas, or even farmed deer, all these biosecurity rules apply just the same. Vigilance is key. But those wild deer we talked about, they still seem like this wild card, this potential source of infection that we just can't control, right? That feels like a gap.
It is. It remains a significant challenge. That interface between wildlife and domestic livestock is tricky. You can't quarantine wild deer. You can't enforce vector control across forests and fields they move freely. So yes, free-ranging wild cervids represent a potential source of exposure for domestic animals that's largely outside our ability to manage directly. It's a persistent vulnerability in the system.
Wow. We have definitely covered a lot of ground. It really is fascinating, this sort of double life that vesicular stomatitis leads. On one hand, often a relatively mild disease for the animal itself, but on the other, because it looks so darn similar to foot and mouth disease, it triggers this massive national level emergency response. And we've touched on the unique situations with camelids officially susceptible, but clinically rare, and cervids widely infected, but apparently mostly subclinical. Real ecological puzzles there.
And even with all we know or think we know, Those critical knowledge gaps are still there, aren't they? Like, what does VSV really look like in llamas and alpacas day to day? Why don't we have more detailed reports? Is it truly mild or just missed?
Mm.
And the deer question, what precise role are they playing? Are they truly just dead ends, or is there something more subtle going on in how they maintain or influence the virus spread that we haven't figured out yet? Every answer seems to lead to more questions.
It really does. So maybe the next time you hear about what seems like a small animal disease outbreak somewhere, woo, take a second. Consider how just one tiny blister on one animal can set off this chain reaction, affecting a local farm's bottom line, yes, but also potentially impacting international trade deals, sparking major regulatory action, and touching on our national agricultural security. Kind of makes you wonder, doesn't it? What other seemingly small details out there in our incredibly connected world might be quietly holding the keys to much larger risks, larger vulnerabilities that we haven't quite acknowledged or explored yet? Something to think about.